guys, welcome or welcome back to my channel. Today we are going to discuss the mechanism of apoptosis. As you might have seen in the recent video that I've discussed the properties of apoptotic cells and the overview how the apoptosis process occur and how it differs from the process of necrosis. Now we are going to discuss the mechanism of apoptosis. Okay, so first basically we will discuss that one thing we missed out in the last video was that how an apoptotic cell differs from a normal cell. It is that when we see a normal cell, as we all know that all the cells are bilayered. So in a normal cell we will see that phosphatidylserine is present on the inner leaflet, and when an apoptotic signal occurs or is received, then this shows a flop moment which occurs due to the floppase enzyme. You might be knowing about flippase and floppase, right? So the phosphatidylserine moves to the outer surface, and hence this acts as a ligand to the receptor present on the macrophage, and thus this macrophage engulfs the apoptotic cell. This phosphatidylserine binds to the receptor on the macrophage. Okay, so the main thing first we will discuss about this is caspase enzyme. Caspase has a very important role in the process of apoptosis. What are caspases? They are cysteine dependent aspartate specific degrading enzyme, right? So what we observe here, why it is named so, that because on the caspase, suppose this is the caspase, on its active site we see that it is containing cysteine, right? And it targets to a protein. Obviously it is a protein degrading enzyme or a protease, you can say. So it targets a protein, and caspase attacks on the aspartate specific site of the protein. That is why it has got the name. C stands for cysteine, asp stands for aspartate, and A stands for protease enzyme because it degrades a protein, right? So next we see, in mammals we have 12 caspases. 1 2 3 4 5 6 7 8 9 10 11 12, right? These are of two types basically. First one is inflammatory or the inflammation caspases. Then we have apoptotic or apoptosis caspase, right? Inflammation means they show swelling if they have been in process or they have been used or they are active. They are 1 4 5 11 12. Apoptotic or the apoptosis caspases are 2 3 6 7 8 9 10, right? In apoptotic also there are two types. They are initiator and executioner, right? Initiator are 2 8 9 10. Executioner are 3 6 7. You might be knowing initiator means to start the process of apoptosis, and executioner means they execute the entire process of the apoptosis. Let me give you a brief overview what happens exactly in the process, right? Look here. First we have an initiator, procaspase. Why it is procaspase? Because it is in inactive form, right? It is in an inactive form. So then comes an apoptotic stimulus, which converts the initiator procaspase into initiator caspase. Thus it becomes active. So this acts on executioner procaspase and convert it into executioner caspase. And this executioner caspase then shows apoptosis, right? This is the brief. Now let's get into detail. What happens in this process? First we see that this is a procaspase, right? It has two terminals, the N terminal and the C terminal, right? So then comes an adapter protein which leads to the binding and helps in dimerization. Dimerize होने के बाद it becomes active. That is, procaspase turns into caspase. Let's see how. See, for example, this is a procaspase. Oh, sorry. And here it is, right? I'll have to erase this part. Okay. Assume this is your adapter protein. This dimerizes it, that is, it brings two units together. N terminal is also referred to as the adapter binding site or the adapter binding domain. Okay. And when we look at the C terminal, it has a cleavage site, right? Cleavage site, we will discuss about this as it comes on further, right? Cleavage site, it comes, okay, as it dimerizes, this C terminal acts on this cleavage site and this acts on this cleavage site, and both of them cleave each other, right? As it cleaves it, it comes upward, like I'll show you here. It becomes like this. Okay. Now this procaspase has become, as it has joined together, it becomes active caspase. Okay. Now it acts on multiple substrates, like it can act on DNA and protein, leading to their fragmentation or apoptosis. Then it can act on the nuclear lamina. It can also degrade the cell-cell adhesion. Then we see that it also acts on the cytoskeleton, as we have discussed in the previous video, that it degrades the cytoskeleton, leads to the cytoskeleton breakdown, which leads to the process of apoptosis. Okay. Now I hope it might be clear that how the procaspase converts into caspases, right? This was the story of initiator, and same happens with the executionary also, right? Now let's move on further. There are two processes of apoptosis. One is the extrinsic one, then we have the intrinsic one, right? Extrinsic, as the name suggests, that an external stimulus might have led to the process of apoptosis, and intrinsic mean an internal signal might have led to this process, right? Okay, so this is also known as receptor mediated apoptosis. Okay. I hope it might be clear till here. We'll discuss both the processes in the next video. If you like the video, please like, share and subscribe. Thank you.